Hello class, welcome to our first lecture video here on the topic of meiosis, which is going to be all about making both sperm in males and eggs in females. Meiosis is the process where we're going to go through two division stages in order to produce haploid cells. All right, so again, sperm and egg are going to be considered what we call gametes. So definition, again, is the formation of haploid gametes, sperm and egg. It is going to be similar but yet different to mitosis as far as comparison goes. In meiosis, we're going to see that there's actually two division stages as opposed to only one, and we'll have then a total of 8 phases. So we're going to see a PMAT1 and a PMAT2. We'll be producing 4 cells instead of 2. And in humans, each one of these 4 cells will have 23 single chromosomes as opposed to 46. And the key thing is each one of these 4 cells will be completely different genetically. All right, you'll never make the same sperm or egg cell again for the entire period of your life. While in mitosis, you're making identical daughter cells every time a somatic cell goes through division. Ultimately, why this is occurring is you are trying to introduce and create variety in your offspring. And where there's variety, it generally increases your overall survival chances, leads to change and evolution over time. If we just look at a quick comparison diagram here, let's say we start with a diploid cell here, so that 2n symbolism is again referring to a diploid number. So we have a total of 4 chromosomes. Regular mitosis would go like this that we've already looked at. You'd see sister chromatid pairs. They would eventually line up individually during metaphase, and then after anaphase and telophase, you'd have 2 cells that each have 4 chromosomes apiece, and again, they're 100% identical to the original. But with meiosis, we're going to go from 1 cell, we're going to go through the first division stage, and we're going to get 2 cells here that are already going to be considered haploid. If you take a look, there's only 2 chromosomes, or 2 X's in each one of these cells, as opposed to 4 that we began with. But we're not going to want sister chromatid pairs or the X's. We don't need duplicate versions of these chromosomes. So a second division stage is needed for each of these 2 cells. So we end up with 4 cells that all have just 2 single versions of all chromosomes instead of 4 like we started with. And if you look, you notice that every chromosome is a little bit different. You're going to notice that we have some blue-red hybrids, mainly red with a little bit of blue hybrids. And that's going to be a result of something that happens in the very first stage of meiosis in prophase number 1. So we're going to look at meiosis 1 here in this lecture. So like mitosis, again, there's 4 stages. For meiosis 1, we're going to have what's called prophase 1, metaphase 1, anaphase 1, and telophase 1. After meiosis 1 is done, our overall outcome will be that we would have taken 1 diploid somatic cell. So this would be in the testicles of a male or the ovaries of a female. You do have diploid cells that would have 46 chromosomes. And that 1 diploid cell will end up producing 2 haploid daughter cells. All right. However, these 2 daughter cells, although haploid, the chromosomes are still in the X shape, the sister chromatid pair form. So we will need to undergo a second division stage, meiosis 2, to eventually split those sister chromatid pairs. All right, so let's begin, and we'll start off with looking at the key differences that occur in meiosis 1, but not mitosis. All right, these next 2 slides are both going to happen during prophase number 1. And the first key thing that happens is a process called synapsis, where the homologous chromosomes, so the versions for a chromosome from both mom and dad, find each other and pair up. So they actually give each other a big hug and embrace. If you pay attention to this little animation down here, you can see that happening. That does not happen in mitosis. So when synapsis happens, the homologous chromosomes or pairs come together. So you can see the creation of homologous pairs, but they can also be referred to as tetrads. So when two homologous chromosomes or sister chromatid pairs come together, like you see right here, you form a homologous pair. We could also refer to it as a tetrad at this point. Tetrad really refers to 4 chromatids total. So if you have 2 X's coming together, you do have a total of 4 chromatids. So now when they embrace, and since DNA is held together by hydrogen bonds, you can actually now end up having an exchange of genetic information occur between these 2 homologous chromosomes. And that never happens during mitosis. So once synapsis occurs and we form these pairs or tetrads, the next unique thing that can occur is something called crossing over. This is when the homologous chromosomes or tetrads will exchange genetic information between each other. And if you look at the 2 animations down below, you can see a whole purple chunk being exchanged with a blue chunk on this other chromosome. All right, and this happens randomly. A whole purple arm could be exchanged with a whole blue. It could be just the bottom portions. It could be the top portions. It could be the middle portions. There's no rhyme or reason to the genetic exchange. Again, we just have 2 different animations showing us that process happening. So you're creating now unique chromatids or chromosomes that have never existed before during crossing over, which is happening again during prophase I. This is where all the variation in offspring comes from, or the majority of it, which again leads to variation and eventually evolutionary change through the process of evolution. So synapsis and crossing over both happen during prophase number 1. Again, when you look at it, we can see the process here. Here's synapsis, and then crossing over occurs. If we do some quick terminology here, up here I have 2 chromosomes. I could also say I have 2 X's, so that would be 2 sister chromatid pairs. Or 1 homologous pair. The blue and the red both contain the same genetic information. 4 total chromatids. Each half of an X is a chromatid, so 1, 2, 3, 4. And we could throw in that one new term here. I also have 1 tetrad. So again, that vocabulary is important once again as we move forward. Next, we're going to hit metaphase number 1. Now, in metaphase number 1, it's the tetrads, or again, homologous pairs, that line up along the center of the cell, just like you see happening here. All right, the spindle fibers are pushing the homologous pairs / tetrads to line up in the middle. This is what we saw in mitosis, but this is not what we're going to be seeing here in metaphase number 1. We don't see the individual sister chromatid pairs line up. We see tetrads or homologous pairs lining up. And then once we get to anaphase number 1, it's going to be the tetrads or homologous pairs that split, not the X's or the sister chromatid pairs. So this is what we're seeing down here in this animation. You can see that the X's don't break down. It's just the tetrads or homologous pairs. So what's not happening here in anaphase number 1 is the chromosome number does not double like it did in mitosis. So again, over here, this is not what you're seeing in anaphase number 1, where the chromosome number would go in this instance from 1, 2, 3, 4 to 8. You're starting off here with 2, 4, 6. And once they split, you still see 1, 2, 3, 4, 5, 6. All you're doing is splitting the tetrads or sister chromatid pairs. If I go back again, just so you can see that one more time. So I'm sending 3 chromosomes to the right and 3 chromosomes to the left. And by the time that telophase 1 and cytokinesis are done, I will have produced 2 haploid daughter cells. If we think of it in terms of humans, that means I would have went from 46 X's in the original cell to now 2 cells that have 23 X's, or chromosomes slash sister chromatid pairs. So the chromosomes are going to still look like this down below, which is what we did not see in mitosis. So we did cut the chromosome number in half, but we still have these sister chromatid pairs. All right, so that's why a second division, or meiosis II, will need to happen in order to take these two cells and split their sister chromatid pairs. So if we look at a diagram, just to kind of finish this out, we start in interphase in the ovaries or testicles. All right, DNA duplication would occur. We get to prophase I. Synapsis happens. You can see homologous pairs or tetrads form. And then crossing over will occur where the blue and red chromosomes, the homologous chromosomes, will exchange genetic information. In this cell here, you would have, if you count the centromeres, you have 1, 2, 3, 4, 5, 6 chromosomes, or 6 sister chromatid pairs, or 12 total chromatids. Each X has 2 chromatids. And I also have 1, 2, 3 homologous pairs, or again 3 tetrads. Metaphase I, they line up as homologous pairs or tetrads down the middle of the cell. Again, left to right or up or down, doesn't matter. Anaphase I, it's the tetrads or sister chromatid pairs that split. So I'm not going from 6 chromosomes to 12 right now. I still have 6. 1, 2, 3, 4, 5, 6. But I have 3 moving in one direction and 3 moving to the other. So by the time I hit telophase I slash cytokinesis I, I'm going to have 2 cells. I have 2 new nuclei starting to form. Eventually, cytokinesis will finish. You can see the cleavage furrow starting to happen now. But we've gone from 1 cell that started with 6 chromosomes to 2 cells that now have 1, 2, 3 chromosomes apiece. So they are haploid, half the genetic information. But since they still have their chromosomes in the sister chromatid pair form, these 2 cells here each will have to go through a second meiosis, meiosis II, in order to split those sister chromatid pairs, and we'll eventually get 4 cells that each only have 3 individual or single chromosomes apiece. All right, and that's what we'll look at as we move through in our next lecture video and look at meiosis II, which honestly looks just like regular mitosis. But again, key differences really in meiosis I versus mitosis. We had synapsis and crossing over occurring. Tetrads or homologous pairs line up as opposed to just individual chromosomes. And then during anaphase I, it's the tetrads or homologous pairs that separate, not the individual chromosomes or sister chromatid pairs.